Hello friends, this is Dr. Anjani, Assistant Professor in Biotechnology. Today we learn about the Haworth and Fischer projections of glucose, a monosaccharide, in session 5. Glucose is also called dextrose. It is a simple sugar, means glucose means sweet, since it is sweet in taste, and it has the molecular formula C6H12O6. It is found in fruits, honey, and it is the major free sugar circulating in the blood of higher animals. It is the source of energy in the cell function, and the regulation of its metabolism is of great importance. Otherwise, we will face many health problems. The glucose is also the important component of many polysaccharides like the starch, glycogen, and cellulose. The starch, which is the reserve food material in case of the plant, the excess of glucose is stored in the form of the starch in the plants. Here it is made up of thousands of linear glucose units. They are placed linearly, one after the other. The another major compound that is composed of glucose is cellulose. So this cellulose is an important component of the cell wall that is present in the plants. Here also we'll find the glucose units which are placed linearly. The animal storage food material is the glycogen. The glycogen is also having the glucose units. And these reserve food materials, starch and glycogen, they act as the source of energy. This is the Fischer projection structure of the glucose. It is having C6H12O6, and as earlier we discussed in the ribose Fischer projection, usually Fischer projection will have the carbonyl at the top and then followed by rest of the carbon atoms. So at C1 we have the CHO aldehyde group, at C2 we have OH towards right side and H towards left side, and at C3 carbon atom you have H towards right side and OH towards left side, and at C4 carbon atom we have OH towards right side and H towards left side. At C5 carbon atom we'll find OH towards right side and H towards left side, and at the C6 you have CH2OH, and here the structure is linear and you will find the functions projected on either side of the carbon skeleton. And we know the last carbon atom, the last but one carbon atom, if it has the OH towards the right side, it is called D sugar. Since here also in glucose structure we have OH towards right side, so it is D glucose. Similarly, so this is the structure of the glucose, but here you have the OH towards the left side. OH towards left side. So it is L glucose. L glucose. And the most of the sugars are D sugars. Most of the sugars are D sugars. And if you put a mirror here, it is the mirror images of each other. It's a mirror images of each other. That is D sugar and the L sugar. So in D sugar, so OH will be towards right side, and in L sugar, OH will be towards left side on the lower carbon atom. That is the last but one carbon atom in the structure of the sugars. This is the Haworth projection structure of the glucose, where you have all the functional groups which are placed towards the right side or towards the downward position of the Haworth ring structure. So whatever you have the functional group towards the right side in the Fischer projection will be placed towards the downward side of the Haworth ring structure. And here the cyclization takes place between C1 and the C5 carbon atom, forming the hemiacetal ring, having the hemiacetal ring, and that will be having the six-membered ring resembling the structure of the pyranose, but in case of the ribose we find the structure like this, because the structure, the cyclization or the cyclic ring formation was between C2 and C5 carbon atom, C4 carbon atom, because it was the pentose sugar. But here it is a hexose. You have the cyclization or the cyclic ring formation takes place between the C1 and C5 hemiacetal with pyranose structure. And in the Haworth structure at C1 you have the OH towards right side, that is towards the downward side of the Haworth structure. At C2 in the Fischer projection you have the OH towards the right side, so you have the OH downwards. And at C3 you have OH towards the left side, it is placed upwards in the Haworth ring structure. And at C4 position you have OH towards right side, that is placed downward of the Haworth projection. And at C5, since there is the hemiacetal ring or the cyclic ring structure formation takes place, so it represents, it is joined with the C1 atom, and to the C5 there is attachment of the C2, the C6, that is CH2OH. So this is the Haworth structure of the glucose. Converting a Fischer projection to Haworth projection, that you learnt in the previous slide. Here at CHO, the first carbon atom is binding to the C5 carbon atom. That's why we have written the O between C1 and C5. So there is formation of the hemiacetal ring with the six-membered ring will be formed, and that's why you have the O between C1 and C5. And OH is downward, so it is on right side in Fischer projection. So here you will be having OH towards right side. So that's why it is shown towards the downward end of the Haworth projection. Sorry, Haworth structure, ring structure. And at C2, OH down, and at C3, OH up because it is on the left side, and on the fourth position, fourth carbon, OH right side, so that's why it is downward. And at C5 there is hemiacetal ring formation, the OH is between 1 and 5, and to the fifth carbon atom there is attachment of the sixth carbon atom, that is CH2OH, so that is placed here. So this is regarding the how to write the Fischer and Haworth projection. Now what when we represent the glucose or the sugars, we will represent them as alpha D or beta D glucose structure, or it is pyranose structure because it is a six-membered pyranose structure. And here D representation we know, if it is towards the lower carbon atom is towards right side, it is D. If it is towards left side, then it is called as L. Then another designation is alpha and beta. This is called the anomerization. This is called anomerization, and the alpha and beta are called as the anomers. They are called as anomers. What are these anomers? At C1 carbon atom, when there is cyclic ring formation between the C1 and C5, we will write OH towards right side or downward of the Haworth, or OH towards left side or upward side of the Haworth structure. Then such type of arrangement of the OH on the C1 carbon atom, they are called as the anomers. That means if the cyclic ring formation with the hemiacetal ring formation with the OH towards the right side, then it is called alpha. If it is towards the left side, then it is called the beta. You're following? So at C1 atom, if the OH position is interchanged, then such type of the isomers are called as the anomers, and they differ in the configuration at C1 atom. This is writing the beta D glucopyranose structure. In the previous slide, we saw the alpha D glucopyranose, where it has the OH towards the right side, that is towards the downward end of the Haber structure. Next, in case of the beta pyranose, here when there is the cyclic ring formation takes place between C1 and C5, the OH is towards left side. That's why the OH is placed upward end of the Haber structure. So such type of glucose are called as the beta D glucopyranose. Here also the OH is towards right side. That's why it is D, beta D. But on the C1 atom, we have the OH towards the left side. So it is beta D glucopyranose. Okay. So in this session, we learnt how to write the Haber structure and the Fischer projection structure of the glucose, and how we represent them as D-glucopyranose and L-glucopyranose on the lower carbon atom. And then again these glucose are represented as the alpha D-glucopyranose and beta D-glucopyranose when they differ in their configuration of the OH on the C1 atom. I hope today you understood the Fischer and Haber structure of the glucose. Thank you.